Okay, so this is my final exam presentation for microbiology. This is Haley. So let's start. Okay, so the first question was, what was your favorite topic? And so my favorite topic was the microbial infection of the human body. So personally, I enjoyed the microbial infection of the body because I enjoyed learning about all of the different infections as well as what caused them, showing how many different types of bacteria, yeast, or fungi there are, and what can happen when something goes wrong. Considering some of the microbes are different things, such as yeast, in the bread or beer are normal and useful in baking. And so, like the yeast that's in urinary tracts are also yeast, but different types of yeast. They show different signs and symptoms, which is so interesting to me. I feel as though all of the information in this course has been applicable to my career in lab and future degree as an MLT. Understanding what causes the diseases and like what we're testing for. My lab might not be able to do microbiology. However, like we collect blood cultures that determines what microbes are in the patient's blood. So this class helped me understand the results from the cultures a little bit better, as well as how they're grown and preventing some of the diseases. I added some of my lab photos to this slide because I felt it was necessary to show that the lab shows the antibiotics from the discs that inhibit the bacterial growth on two different types of bacteria. This experiment showed that there was the tetracycline, sorry, I don't know how to say that, was resistant for both bacterias and was more resistant to the bacterial growth than the bactitracin was. So this lab helped me understand on how antibiotics work in the body, or for when someone is taking the antibiotics to reduce the amount of bacteria or foreign growth that's happening within the body. And understanding that yes, both bacteria are helpful to inhibit the growth of both E. coli and the S. epidermis, epimentis, I don't know how to say that. However, the tetracycline is a better bacteria or better antibiotic overall to inhibit the growth of both samples. And then again, I showed my results. So I have my agar plates as well as this. And then there was a picture up there, but I don't know if it's not just loaded. So if I had an unknown microbe, I would start out by determining if this microbe was prokaryote or eukaryote. I can do this by looking at the nucleus of each. So if one has a nucleus, it's a prokaryote, and if the nucleus is not there, it's a eukaryote. Then diving deeper in it, if it was a prokaryote, I would determine if it was bacteria, archaea, or eukarya. Bacteria will show different types of shapes, such as spherical, rod, or spiral, which can determine what type of bacteria the microbe can be. This would have to be looked under a compound light microscope after the bacteria has been put onto a slide and stained. Some of the stains that can be used are gram stain, India ink, or crystal violet. Depending on how the bacteria colors with the stain, which can grow gram positive or gram negative. The archaeogrobes have flagella that produce by binary fission, and a key characteristic of that they can grow in extreme environments such as hot springs. They also can be in a variety of color. The eukarya includes the kingdom fungi, and fungi have spores which are produced by budding, so I can identify the fungi by looking at it the way it grows under a microscope. And then on the other hand, viruses can be determined by noticing that the viruses cannot produce without the host, and that the viruses can be determined by what kind it is, by whether it's a naked virus or an envelope virus. Also determined by the type of nucleic acid within the virus. And then I just added a bacteria versus virus photo just to show. And then, this one was talking about vaccines. Talk to someone if they didn't believe in vaccines to show them what could be the benefits. Okay, so I would start off the conversation by defining what a vaccine is, as well as the different types of vaccines are given. Then lead into the types of immunity this can give you, as well as explain how the body breaks down the vaccine in the body, and lastly, why it is important to get yourself as well as your family vaccinated. So then I'll go a little bit more into that. So this definition is directly from biologyonline.com that I've sourced on the page. So it says a suspension containing live and modified or killed microorganisms or their toxins or tumor antigens, which is when administered into the body, stimulates the body's immune system to produce antigen-specific antibodies. So the antigen in the vaccines are weakened, so therefore bits of the genetic or the exterior surface of the antigen in the vaccine. Also, the antigen in the vaccine is treated to make sure that it's not toxic to the human body. There are vaccines that are live that need at least 2 vaccinations to become effective, and non-living vaccines that require 3 vaccines, and when you will have boosters with that. So live vaccines are meant to last a lifetime. These vaccines include like chickenpox and MMR, which is measles, mumps, and rubella. The non-live vaccines are the protection fades over time and booster needs later. However, these vaccines are good for people with a weakened immune system. Non-live vaccines include DTaP, which is diphtheria, tetanus, and pertussis, which is whooping cough. Babies can get these vaccines at 2, 4, 6, and 18 months. Children will need a booster by the time they enter school. Adults should get tetanus booster every 10 years, or if one's pregnant, with every pregnancy. There are different types of immunity with vaccines, such as natural immunity. Vaccines include passive immunity. Natural immunity is from getting infected from the disease-causing organism. Vaccines include immunity is getting from exposed to the pathogen from said vaccine. Passive immunity can be from mother's antibodies passing to the baby in utero, or getting blood from humans or animals. Vaccines work as when a person gets a vaccine shot in the body, the immune system breaks down the microbes and form bodies, which in turn learns about the microbes so they can render how to fight better next time. It's like an instruction sheet. So the next time that the microbe comes in contact with the body, the immune system knows exactly how to deal with it. And then lastly, vaccines are helpful in many different ways. It saves many lives over the years, and vaccines are a great way to take protection caution with your family against serious illnesses. Vaccines are closely monitored to be safe to receive, and vaccines are required for work, school, and probably other activities that you have in your life. So then we'll go on to the relationships that humans have with microbes. So the gut microbiome is a positive relationship between the human and microbes. The gut microbiome is in the intestinal tract, in your stomach, small intestine, and large intestine, and your colon. They break down fiber, which is not digestible by the digestive tract, and help you pull nutrients that you might not have been able to without them. So babies will get their first gut microbiomes from the vaginal deliveries or breastfeeding or their diet later on. A disadvantage of gut microbes, if they're not in the ideal state, they can cause many problems, such as bacterial infections that are in the GI tract called C. diff or H. pylori, as well as causing inflammation and a flare of one's autoimmune disease. This is called dysbiosis. I never knew how to say that, which means an unhealthy gut microbiome. That leads to a deficient bacterial growth of bad bacteria, which is also the loss of bacterial diversity, which is needed. Other problems with gut microbes can cause infections, small bacterial overgrowth, and inflammatory bowel disease. Another positive microbe with human interactions could include the growth of penicillin, which helps to fight gram-positive cocci and gram-negative cocci bacteria in the human body. Penicillin grows from the mold Penicillium. Penicillin breaks down the cell wall of the foreign body and does not harm the good cells. This medication is used to treat meningitis, syphilis, and sore throats, or throat infections, I should say. A disadvantage of the microbe can include an adverse reaction, which is nausea, vomiting, seizures, muscle pain, and hypotension. Also skin rashes. A disadvantage to microbes to humans are parasites. Parasites feed and grow within the human body. However, they are called ectoparasites and are parasites that live outside of the body. That includes fleas, head lice, mites, or ticks. These parasites infect humans through blood while also feeding on the human's blood. They can come from sexual contact, being in areas with many parasites, and infrequent bathing. On the inside of the body's helminths can live. There are parasitic worms that live in the GI tract. These can be from 1 millimeter to 1 meter in length, which is crazy. The symptoms include increased appetite, abdominal pain, and skin rash. These worms can come from uncooked meat, unwashed vegetables, and unpasteurized milk. Depending on the parasite, a patient might get a stool culture, colonoscopy, blood test, or an MRI. These can be treated, some can be treated with antiparasitic drugs, antibiotics, or shampoos. There's really no advantage to the human from the parasite. However, the parasite's point of view is a positive one because they can have a host that they can survive in until they want to relocate to a new host. So then lastly, a negative interaction is urinary tract infections, which is another disadvantage to microbes. The UTI forms in the urethra and goes into the bladder and causes bacterial growth. Symptoms of a UTI include burning when urinating, smelling urine, pelvic pain, and urine is cloudy or red. Infections of the bladder include E. coli. Sex can be a contributor of the infection, as well as the length of the area between the anus and the urethra for women because it's so short. Infections of the urethra can include infections caused by sexually transmitted diseases as well. These can be herpes, gonorrhea, and chlamydia. The risk factors for getting a UTI include female anatomy, sexual activity, types of birth control, menopause, and catheter use. One can get permanent kidney damage from a kidney infections that's come from an untreated UTI. To help with lowering your risk from a UTI, include drinking water, drinking cranberry juice, and going to the bathroom after sex, as well as changing your birth control. Again, there are no really advantages to the human body for the UTI, but for the bacteria, it has the ideal temperament to grow and thrive in. So it's both of the disadvantages really aren't for the greater good of the human body, I should say. There are many applications of the microbes that are around us. So I included in creating food. With yeast, it ferments the sugars and carbon dioxide that helps bread rise. With milk, there's a lactic acid in the bacteria that is in buttermilk, sour cream, cheese, and yogurt. The lactobacillus in the microorganisms in pickling. Curing meat also has a lactic acid bacteria. which is in summer sausage, pepperoni, and salami. The aspergillus is in the fermentation of soybeans and wheat paste, which becomes soy sauce. And then there's acid bacteria in wine, apple cider vinegar, and dilute ethanol. The Bacillus thuringiensis helps to kill insects and pesticides. This bacteria has a toxin that is directly affecting certain types of insects. Another one could be treating sewage, which includes the primary, secondary treatment, as well as the denitrification, which also helps with the algae in the rivers and the stream when it's let go. A compost pile is broken down by microorganisms and the heat-tolerant bacteria called thermophiles. The temperature of the compost pile while being broken down increases as well as the carbon dioxide it increases. So when the compost pile has been fully broken down, it can act like a fertilizer to soil around it, or if it's moved to a crop. Soybeans, peas, and beans are worked with nitro-fixing bacteria, which is a rhizobium? I don't remember. That can help with root cells and help with nodules so that they can produce more nitrogen compounds that are given to the plant so that the plants will give them shelter. It's kind of a give and take relationship. And then I just wanted to end it. There we go. With all my sources, I did do a lot of research for all of these. I did use some online, and then I also used some of our lecture material, as you can see. So that is it. Thank you for my presentation.